and this is called antigenic drift. We have to remember that this antigenic drift, this process, is much slower than what happened during antigenic shift. In Bangla, forget about that. So, antigenic drift is much slower process. And because of this antigenic drift, okay, what I was trying to say, that if we like to compare the shift and drift, we can say that Shakrar tuk kamarer ekgha. Okay, so this drift is tuk and shift is practically ekgha. Now, because of this antigenic drift, every 2 to 3 years, vaccine strain need to be updated, and we need to take vaccine again. H2N2 Asian flu pandemic and 1968 Hong Kong flu pandemic both occur because of human and avian-like virus reassortment. In the year 1957, seasonal H1N1, which was actually descendant of 1918 H1N1 virus. At that time, in 1957, it was a seasonal flu to us. And that virus was reassorted with a avian H2N2 virus and took PB1, HA, and NA from that avian virus and caused that 1957 Asian flu pandemic. And then, with time, this virus became the seasonal flu of H2N2 seasonal flu for human population. And then again in 1968, that time it was a seasonal flu of H2N2. That H2N2 seasonal flu again reassorted with another H3 avian virus and took PD1 and HA from that avian virus and caused that 1968 Hong Kong flu pandemic. And then again with time, that virus became a seasonal H3N2 virus to us, and even today that virus is circulating within us. In 1977, there was a mild pandemic in Russia. It was that was entirely that 1950s H1N1 virus which outbroke to human, and because that sequence was very close to that 1950s virus, scientists believe that it might be a laboratory leakage, unintentional laboratory leakage from somewhere in Russia, and caused that mild pandemic. And from then, that virus is continuing within us as a seasonal flu H1N1 until 2009. In the year of 2009, we know what happened, and I will come to that later. But during 2009 pandemic, that new H1N1 kicked out that virus from human population. So this is the story of pandemics 1957 and 1968. Now the question remains: what happened during 1918 Spanish flu pandemics? And that was a very devastating pandemic in human history. So how we can get more insight of that event and virus during that time? In 1951, a PhD student, Halkin, heard about 1918 Spanish flu virus while attending a seminar on that topic. And then it came to his mind, like how we can collect that virus so that we'll know far better in depth about that devastating Spanish flu virus. But from where and how we can get it? Mainly with his initiative, a research team from Iowa University went to Breivik Mission Village in Alaska. Why Alaska? Because Alaska is snowbound and about 85% of Alaska covered with permafrost. Permafrost is a frozen layer underneath the soil surface. Furthermore, it was reported that in Breivik Mission, many Alaskan people died because of 1918 Spanish flu. Therefore, in the graveyard, there may have some chance of getting live 1918 H1N1 virus. So after getting permission from local authority, they dig the 1918 victims' mass graves and collected samples. Unfortunately, no virus had been isolated. In the meantime, biological sciences advances a lot. During 80s, we got PCR technology, and in the year of 1995, Taubenberger initiated a project to isolate viral RNA from processed viscera samples of 1918 Spanish flu victims, which was stored in US Army facility. If it is possible to isolate viral RNA, then sequence can be done and we can understand better about the 1918 virus. In fact, they did so. And in March 1997, they published a paper in Science where they described that 9 different fragments, not complete gene, but 9 different fragments from HA, NA, NP, and M genes. So it was a big moment for influenza researchers. And Haltin, at the age of 72, got to know about that publication, because it was really surprising. Like we can do it, and we can get some information from 1918 influenza virus. So then he wrote a letter to Taubenberger expressing his desire to join with him. Taubenberger agreed, and agreed. And then a second time, a research team went to same place, that Alaska Breivik Mission village, with the leadership of that 72 years old, that Haltin, leading that team. And then they collect samples. This time they collect sample with the intention to isolate RNA, not to isolate virus. That because they know that they already failed before. So this time they collect samples, and then from these samples, as well as more samples purified from viscera samples, altogether, finally they sequence the complete genome of 1918 virus. And then we know that it was not a reassortment, rather than it was an intact H1N1 avian virus that jumped to human and caused that devastating pandemic. Of course, nothing happened overnight, and then research on 1918 virus was going on. All these things are well described by Potik Go on 26th July, Anandabajar, Ruibashario. You can read it if you have not already done so. It is well written and worth to read. Now coming back to that point, like already I have told that Taubenberger published their first paper on 1918 virus in the March 1997 and energized influenza researcher a lot. Just after 2 months, in May 1997, a report came from Hong Kong that a 3-year-old boy was admitted to the hospital and later died from influenza pneumonia and other complications, including the multi-organ failures, etc. Later, infection was confirmed in 18 individuals, and 6 of whom died. Sequence analysis revealed that this infection were acquired by humans directly from avian without involvement of any intermediate host. And of course, no human-to-human transmission were reported. That HPAI, hypoavian influenza, we know about that virus long before. Since 1959, these HPAI viruses were causing havoc to chicken, duck, or turkey farms. However, human infection and, more importantly, death was unprecedented. Here I must explain a little bit on that. What is HPAI? What is high path avian influenza? The HA protein binds to cell surface receptor, that is sialic acid receptor, and virus entered the cell via endocytosis. And then at the late phase, that low pH, late endosome, that viral membrane, and shown in the Turkish color and then green color, that endosomal membrane, these two membranes have to fuse to make a passage for genomic material to release in that cytosol. And this thing, HA protein is responsible for that actual membrane fusion. But however, to do so that membrane fusion, HA must cleave to HA1 and HA2, which will express the fusion peptide, and then that membrane fusion will happen. Most HA cleavage sites are LPAI-like, low-path avian influenza, and those HA can be cleaved only by trypsin-like protease, which is available only in our respiratory system. And so virus can only grow in our respiratory system. In contrast, that HPAI, that high-path avian influenza, they contain extra multiple basic residues like arginine or lysine, and that makes that HA, it can be cleaved by so many different kind of proteases which are readily available in different kind of cells. Therefore, HPI viruses, potentially, they can grow other than respiratory system. And here we have to mention that chicken death rate due to HPI virus is almost 100%. However, we are talking about Taubenberger's research on 1918 virus and HPI infection and human death in Hong Kong. While all these things were happening, in 1999 August, we got another very important paper in PNAS describing reverse genetics of influenza A virus. That means generation of influenza A virus entirely from cloned cDNA. Just 3 months later, we got another paper on Journal of Virology describing the similar matter. Both the groups, they used a system that 8 plasmids where they cloned each gene in a plasmid under human POL1 promoter, and which will express that negative sense viral RNA. Then first group use another nine plasmid to express 9 proteins, that influenza proteins under same promoter. In contrast, the second group, they use 4 plasmids only to express only 4 proteins. That is PB2, PB1, and PA, which makes the polymerase complex, and NP. I told before that inside the virus particle, that RNA exists as a BRNP complex. And to make the BRNP complex, the necessary thing is that RNA, viral RNA, NP, and then 3 polymerase proteins. So when this viral RNP complex is formed inside the cell after DNA transfection, then that cell will behave like an infected cell. And then viral genome replication will begin, and finally we will get the progeny variants from there. Next year, again we got another paper on PNAs describing the same reverse genetics, but in this case they use only 8 plasmids. 8 genes was expressed, that viral RNA was expressed similar way like before other 2 papers. They express negative sense viral RNA under human POL1 promoter. Why human POL1 promoter? Because that promoter, that transcript length is very precise, so that we can maintain the viral RNA length, no changes there and all those things. And really cleverly, smartly, they use that CMV promoter in the other end in opposite direction to express mRNA, so that all the proteins can be synthesized inside the cell. So same way, when BRNP complex will form, then it will behave like a virus infected cell, and we will get infectious virus particle. So this is this reverse genetic system. That means we can make infectious virus particle from plasmid DNA. So we can prepare our plasmid DNA in vitro, and then we can transfer those plasmid DNA to get a real infectious particle. And that is a big achievement for influenza research. Before, we can only try to correlate, like we do the sequence of that virus, and then we do some animal study, virulence study, and all those things, and we then correlate with field data to understand that which gene or which amino acid in each gene they might be contributing to that higher virulence or lower virulence. But reverse genetics allows us to pinpoint like exactly which gene and then which amino acid is responsible. We can mutate those in vitro and then make that virus, and then we can study their virulence. So that reverse genetics is a very big step for influenza virus research. Then using that reverse genetics, the 1997 Hong Kong H5N1 virus was further. We studied that virus, we characterized that virus, and we know that from that study that high cleavability, that is the one factor that HA can be cleaved in so many defined cell with so many defined kind of protease, so it can replicate in defined organ other than respiratory tract. That is the one criteria. But beside that, there's another important factor. It is that PB2 6 to 7 lysine. In fact, PB2 6 to 7 position lysine, we can see it is mostly in human viruses, and avian viruses, they contain at that position that glutamic acid, that E. So using that reverse genetics, first they choose two viruses. One is Hong Kong 483, which is highly pathogenic in mice. Another one is 486, that is much less pathogenic in mice. If you see here these numbers, lower number means higher virulence, because in this case 1.8 means about only 2 infectious particles can kill 50% of mice. But in case of 486, you need 10,000 infectious particles to kill 50% of mice. That's why I say that when the number is low, that means it is more virulent in mice. So by swapping the genes, if that 8 genes from high path and 8 genes from low path, they swapped and then they make a series of viruses using reverse genetics, and then they figure out that PB2 gene is also playing an important role for high virulence of 483 virus. That part I didn't show in this slide. But further, they make a chimeric protein swapping region from PB2 of each viruses, and then they pinpoint a small region which is mostly responsible for higher virulence of that 483 virus. And finally, they mutate that avian-like virus to human-like lysine to glutamic acid in high-path virus and glutamic acid to lysine in low-path virus. And then they find out that virulence also switching accordingly. And in this way, finally, we got to know that at PB2 6 to 7 lysine is very important for high pathogenicity of Hong Kong H5N1 viruses. On the other hand, Taubenberger also continuing his research on 1918 virus. And after reverse genetics genetics is available, they finally rescued the infectious 1918 virus. That is another big step of influenza virus research, because nobody ever thought that we can make that virus, that 1918 virus, and then because of that reverse genetics, now they can synthesize the DNA and then they can make that virus. Also, that is done because they also completely sequence that virus from that sample from Breivik mission as well as from viscera samples. Then after making a live virus, not live virus, but infectious virus, and then finally we find out that NA gene was responsible for the cleavage of HA in absence of trypsin. That means that is NA gene is playing very important role for high pathogenicity of 1918 virus. How they did it? Like without added trypsin, I told you before that LPAI virus, that HA can only be cleaved by trypsin-like protease, and that trypsin-like protease only available in our respiratory system. So other cells, other kind of cells like say dog kidney cells, which mostly we use for plaque assay or virus application, influenza virus application, those type of cells, they don't produce any trypsin-like proteins. So to replicate the virus on that type of cells, we normally use exogenous, we add trypsin to that cell so that virus can replicate. And that's why those virus only can produce plaque if we add trypsin. Here you can see that two low-path virus, they are not producing any plaque in absence of trypsin. However, 1918 virus, that HA sequence told us that it is a low-path-like virus. It don't contain that multivic basic residues at the cleavage site, but still we can see that that virus can produce plaque without any trypsin. And after swapping genes, they finally find out that Na is somehow doing that job. Actually, later on, we find out that Na is protein is accumulating plasminogen, some other kind of enzyme, and enhancing that type of cleavage of H. So this reverse genetics, actually, I'm talking only about two of this major work, but there are endless examples of such works, very good work using that reverse genetics. But beside that influenza research, influenza virus also help us for that reverse that influenza vaccine virus production. So, but before going to that, how in that reverse genetics can help us to get influenza vaccine virus production, first I need to talk a little bit on that influenza vaccine. Currently, two subtypes of influenza HA viruses circulating. Like one is descendant of 2009 pandemic H1N1 virus. Another one is H3N2 seasonal flu, which is the descendant of 1968 Hong Kong pandemic. And also, we have that influenza B viruses, and for decades, that B virus HA also segregating their antigenicity. They are in two different groups. One is called Victoria-like, another one is called Imagata-like. But the dissimilarity among these type of HA in B are not that much far, as we see in case of influenza A, H1HA or H3HA. That's why we have to keep these two viruses in vaccine, but sometimes we use only one kind of influenza B viruses because they are not much different. So that type of virus, we call it is trivalent vaccine. And of course, when we use two different lineages of B viruses, it is called quadrivalent vaccine. The process of selecting these four viruses, it is a gigantic job. It is a worldwide surveillance, actually conducted through World Health Organization's Global Influenza Surveillance Response and Response System. So, and they are doing that job since 1952. Worldwide, there are 140 national influenza centers. In India, we have 3. One is National Institute of Biology in Pune, another one National Influenza Center, Kashauli in Himachal Pradesh, another one is in Mumbai. That is called Hapkin Institute or something in Mumbai. There are 6 collaborating centers worldwide, that is Australia, Japan, China, UK, 1 each, and we have 2 in USA. St. Jude is one of them, and the other one is CDC. And there are 4 essential regulatory laboratories: Australia, Japan, UK, and USA, 1 each. So data is coming from throughout the world, from all these 140 different laboratories, and then it is coming together, and then we around selection. I can show you one of such data, how much data we are gathering year round. Here it is shown that 2013-14 season, 1 year length of that surveillance data, and you can see we all know that flu are prevalent in colder months. That we know, but in case of northern hemisphere as well as in southern hemisphere, in both cases, influenza A viruses are dominant over influenza B viruses. A viruses are shown in greenish colors, and that orange color you can see influenza B viruses. In case of influenza A, within that type of influenza A virus, that H1N1, that light green, was dominating in northern hemisphere, and in contrast, that H3N2, that deeper green color, that was dominating in southern hemisphere. Twice a year, vaccine strain selection meeting happened. In February, we select vaccine virus for northern hemisphere, which will be released on during that September, October, and also another meeting happened during September to select vaccine strain for southern hemisphere, and that vaccine will be released during February. February. So you see that meetings are happening February and September. Those are very nice months, very good weather. But it is not happening because of good weather and it is fun to do this that meeting on symposia. Rather, we choose those months because of manufacturing limitations. Because manufacturing facility, they need some time. After we select, then we send that viruses to different manufacturing facility, and then they will start their vaccine production, and they need at least 4 to 5 months at least to make that a vaccine. So depending on the season, when season begins, like southern hemisphere season begin during February, and northern hemisphere that flu season begin during September, October. So to keep ready that vaccine during that time, before that time, we have to select at least 5-6 months before. Now, what happened during this 6 months? Sometime, like what we can see during this time, what type of virus is prevalent and moving around the world, and we are predicting that okay, this virus we can see this strain is, or this type of strains are prevalent. So we recommend okay, go for this strain for H3N2 or this strain for H1N1. Some years it happened that during that 6-month period, influenza virus changed its track, and which one looks like may not be prevalent or dominant during next season, but they become dominant. That is called vaccine mismatch. It happened sometime. Like we are making a vaccine with some strain, but finally find out that the prevalent virus is not similar to that vaccine strain what we choose. Though this is not like parrot reading. We do collect a lot of data, lot of sequence data. Along with sequence data, we also collect their antigenicity and that virus efficacy. Lot of factors we consider. Of course, there's one factor is prediction model, and that prediction model is still in very childish condition. But that is the only one factor. All other factors were big factors. They are like how, say, we collect a virus and then we raise antibody against that selected virus and test it for several field samples to check that which antibody is inhibiting most of the virus. And that way we can pinpoint that vaccine strain. So all these different factors are involved. But still, sometimes we miss. Now coming back to that manufacturing process, influenza vaccines strain are nothing but a reassortment virus between a high growth master strain we have in our laboratory and a circulating strain. So we co-infect these two viruses in egg, and then I told you before that when we infect two different kind of virus, there are 256 possibility of genotypes. So our next job to select the appropriate one. For selection, there are three criteria. First criteria, that new virus, which one we are going to select, that should contain HA and NA from circulating strain, because we are preparing vaccine against that circulating strain. And second criteria, it should contain minimum number of gene from circulating strain, because circulating strain are not well characterized. So we don't want that it will carry more and more gene from circulating virus. And third criteria is it should grow in high titer in eggs. That's why we use that high growth master strain, which is, and also I have to mention that when we use circulating strain H3N2, then we use master strain H1N1 PR8, and when we use circulating strain H3N2, H1N1, then we use master strain H3N2. That means circulating strain and master strain, their antigenicity should not match, and that help us during this selection process. And I also need to mention that beside that inactivated vaccine, there are live attenuated vaccine also available, and that vaccine was made very similar way, except the master strain is now, it is not that PR8 or XRT1, it is some cold adapted attenuated virus, because we don't want that live vaccine virus will bother you. So it is a very attenuated and very low pathogenic, and it is cold adapted. Cold adapted means it can't grow above 35 degrees Celsius. And live vaccine, we always administer as a nasal spray. So when you spray to your nasal spray, after nasal spray, it can only grow to your nasal turbinate. And because if it goes inside, then temperature will rise, and after 35 degrees Celsius, that virus can never propagate or replicate. So that is the criteria for live vaccine. So both that inactivated vaccine as well as live vaccine, we can also prepare that vaccine in cell culture. I've shown here the egg as a preparation or propagation system, but we can also use cell culture. So whatever it is, whether it is inactivated or live propagated, live attenuated, or whether it is produced in eggs or in cell culture, we can avoid this co-infection and selection procedure by using reverse genetics. So we have already cloned that PR8 genes, and then using that 6 plasmid containing 6 internal genes from our master strain, and then we clone HA and NA from the circulating strain, and then 8 plasmid together when we transfect in a cell, and then it will produce the exact that vaccine strain what we want. So that way we can make our tailor-made vaccine using reverse genetics. So on the other side, beginning, like I told you before, that first human outbreak of high-path H5N1, we saw it in 1997 in Hong Kong. And then beginning from 2003, we have seen so many outbreak of a poultry outbreak from different countries in the world, and also some sporadic human infection and death. From 2003 to date, there are 455 people died among 861 human infections reported to World Health Organizations. These numbers looks nothing today's light because of this coronavirus, but this is just sporadic outbreak. If that virus can get somehow some kind of adaptation to human, and because this virus is high path property, that means it can propagate other than other organs, and we know that it can cause about 50%. You can see from here more than 50% death. So somehow if that virus can get more adaptation in human so that it can transmit from human to human, then it can also cause a devastating pandemic. So As a pandemic preparedness, using that reverse genetics, we are making PR8-based H5N1 vaccine every year. We are making that seed virus. It is only seed virus, no vaccine. So we are making seed virus every year and stored it somewhere so that in real need we can save 2 months. Because if we keep ready that vaccine virus, then from there, if we start production, vaccine production, we can save 2 months. While we are waiting for coronavirus vaccine, now we know that saving 2 months is really a big deal. In the meantime, while we all are focusing on high-path avian influenza as a threat for human pandemic, influenza pandemic, in April 2009, human outbreak of a new kind of H1N1 virus was reported from California. And finally, in June 11, World Health Organization declared a pandemic. Sequence analysis revealed that it contained genes from 4 defined kind of viruses. In fact, since 1998, sporadic outbreak of double or triple reassortant North American swine virus was reporting here. Those virus was made with 3 defined kind of viruses: classical swine, North American avian, and human. They reassorted and then they make either double or triple reassortant swine viruses, and also some sporadic human infection of such viruses were also reported. But there was no report of human-to-human transmission. But during early 2009 or late 2008, one such H1N2 triple reassortant North American swine virus and then mixed with another Eurasian avian-like swine virus, and then they formed that H1N1 2009 pandemic virus. And it kicked out circulating seasonal H1N1 virus and established itself as a new seasonal human H1N1 virus. One thing I must mention that after April 2009, there are no relation between this virus and swine, and it transmits efficiently human to human. And that's why it established as a seasonal flu. Now, if we look back the chronology of that 2009 pandemic H1N1 virus, first case was reported from California in April 15, then WHO declared the pandemic on June 11, and clinical trials of new vaccine began on July 22, and FDA finally approved the vaccine to use in US on September 15. That is total 5 months. We have to remember that there was not much safety concern because this virus was made in a well-established vaccine platform, that is inactivated vaccine, which we are using for last decades and decades. So it was well-established method, and its safety pattern, we know that what it can do. So there was not much safety concern for that virus. And just to incorporate that one component, that H1N1 pandemic virus in the vaccine preparation, it took 5 months. Now, in case of coronavirus, vaccine platform is not known. That means inactivated virus or the spike protein itself, or it will be expressed with DNA vaccine or as a mRNA vaccine. So with which platform it will work best, we don't know yet. And also, we don't know that which way the safety will not be compromised. And we never use any vaccine against corona for human. That's why, so altogether, it will take time. If we can get it by this year, that will be a big achievement for corona researchers as well as who are involved in vaccine production. With all this, I should stop here. And what is going on in influenza research, or in which direction influenza research is heading on, I would like to talk about all this on another day. Now it is time for questions and discussions. Thank you all for listening.
Thank you, sir, for this enlightening presentation. And yes, this is the time for question answer. And I ask the participants on Google Meet, if you have any question, you can raise it to sir directly. Dear participants, if you have any question, you can raise it to sir directly. If you have any comment or want to discuss anything about the topic which sir shared with us,
I saw
please
one question.
unmute your microphone and you can raise your question. Yes.
There are two questions I could see in the chat box. If somebody can read it out, that will be better. I see two questions in the chat box. So the admin, you can read that out. Somebody called Sonjib Das asked, how research on Spanish flu influenced corona research?
Okay. Not directly Spanish flu, but flu research as a whole, all the knowledge we are gaining from reverse genetics, and currently we are working with the universal vaccine for flu. So that universal vaccine, we are using different platform. What I told in that talk, that DNA vaccine, mRNA vaccine, or different kind of platform we are using for influenza vaccine. Because current influenza vaccine, we have some limitations. Every year we have to get that vaccine because of it is not universal. Means it can't use for different types of different subtype of viruses. So a lot of research is going on universal influenza vaccine, and a lot of different platform of vaccine preparation we are using. So all those informations are also we can use for corona vaccine production.
Sir, there is a question from Enar Sharaf. Is there any genetic markers to differentiate Asian human and avian influenza A viruses?
Question again, Asian influenza and?
Avian influenza A virus.
Yeah, of course. Because the sequence is totally different. Like what we do, so we sequence a gene, for example, HA, and then we try to match, align those sequences, and from that match, we can make a tree, like which way that gene is changing. And when you make that tree, if I have some tree in my computer, I can show you that there's big lot of different markers. One marker I told already, that PB2 6 to 7 position lysine, it is very specifically for human viruses, and that glutamic acid at that position, it is for avian viruses. There are so many other markers in the different genes, and as soon as we get the sequence, we can tell that whether it is a human virus or it is an avian virus.
Thank you, sir. And I think we don't have any question more. So thank you, Dr. Borman, once again for being with us and joining with us and sharing such information on such a prevalent topic. So all the best to you, and take care, sir. And now we have reached at the concluding part of our program of this international webinar. So I would like to request Gaur Gopal Roy, Head of the Department Mathematics, P.R. Thakur Government College, to deliver the vote of thanks and conclude this session. Thank you all.
Good evening. Good evening, everyone. Thank you. I passed Professor Somor Chattopadhyay for organizing this seminar. We have two beautiful lectures today from two different corners of the world. First, Professor Surojit Chattopadhyay. I have learned a lot on nanoparticles, its uses in bioimaging, electronics, and medical applications, even in medical therapy also. We have also an outline how the nanoparticles are manufactured. And second, Professor Subroto Bormon, we learned that even a person who train for do physics can also do research in virus, and from very good presentation we learned how the virus research are doing since the beginning of this century. I thank you once again for the beautiful presentation. Thank you all.
Feedback link will be sent in the registered email ID. Participants who are registered in today's seminar, feedback link will be sent to their registered email ID.
So thank you all the participants. Now it's the time to leave the platform to see you once again. And thank you. Good night. Bye.